So this morning, we'll be starting off with Melissa Skala. See, I practiced her name and then I screwed it up. She comes to us from Morgrid Institute for Research, where she's also a professor of biomedical engineering at the University of Wisconsin-Madison. Hello, Melissa. She did her PhD at Duke in biomedical engineering, and then followed up with her postdoc also at Duke. And before coming to her current position, she was an assistant professor of biomedical engineering at Vanderbilt. And so she will fill us in on her new work where she's been developing label-free optical imaging technologies. All right, Melissa, I'll turn it over to you. You can go ahead and share your screen.
There we go. Oops. Well, now it's not letting me share it, so that's great. Let's see. Of course, we practiced this 20 times, before, and let's see. Now I'll try it, see if it'll let me share it now. Oh, there it is, yay! Okay, so can you see me?
Yes, your screen's up, I can hear you.
Super, thank you. So I was just telling Holly and Feather how impressed I was that they moved this whole thing to virtual so quickly. It's a massive undertaking, so I really appreciate that. This is definitely a highlight of my year. And actually what I try to do every year at this conference is give a whole new talk. And that's a challenge I give myself every year. And this year, I think I have challenged myself the most out of all the years I've come here, because I'm talking about cardiovascular disease, which I've never done, stem cells, which I've never done. And so I'm super excited to share this work with you. I always try to give all unpublished work, and that is certainly the case today. This is super duper new, so thanks. So it's actually kind of ironic. I haven't talked about cardiovascular disease. It's clearly a worldwide burden. And I know there's lots of ways to prevent cardiovascular disease, but we still have this need for better treatments. And the reason we need better treatments is we don't understand cardiovascular disease very well. And we also need novel approaches to develop new treatments. So I think this really requires a whole new thinking about the disease and about how we can solve it. So one way, I'm a firm believer that we should really study human disease. So most of my lab does cancer research, but it's clear in cardiovascular disease and in many different diseases that the human condition is much different from those in animal models, and that humans differ from each other significantly. So we need to understand the baseline normal distribution of cardiovascular function in humans, and then we need to understand the spectrum of disease that we see in humans. And so one way to do this is to use human pluripotent stem cells. And these are great because we can differentiate these cells into many different organs, including cardiac muscles. That's what I'll be talking about today, is cardiovascular disease. So the vision here is that we take a cell from any patient, a normal patient, a disease patient, and that's the beauty of this. We can then start to model human disease. We can take that somatic cell, reprogram it in vitro, and then differentiate into any organ of interest. So today I'm talking about cardiovascular disease. We're going to talk about cardiomyocytes, which are the muscle cells in the heart. And what we can do then is in vitro modeling. So importantly, we can start to really understand human disease, manipulate it, and look at it in the lab. We can use these models to discover new drugs, and we can use these models to discover how people develop developmental cardiovascular disease. So congenital heart disease, for example. And another avenue actually for these stem cells is we can use them themselves as treatments in regenerative medicine. So we can grow these cardiomyocytes and use them in humans to improve their cardiac function directly. So there's both in vitro and in vivo uses of these human pluripotent stem cells. So this is great. What we're going to have now is a source cell, which is from any patient. We can take their cells and reprogram them into stem cells, and then we can differentiate them into whatever we need. So in our example, that's a cardiomyocyte. So we'll have a population of these cells, and we'll be processing these cells. And throughout this process, inevitably, there is heterogeneity. So we start initially with a heterogeneous population. And as we differentiate those cells or reprogram those cells, there will be additional heterogeneity. So this screams for a need for quality control. Because we have variable quality, we want to be able to pick out those cells that are functional, that will help the patient, and discard those that will not. So what I'm talking about here is what we use in biomanufacturing, and we call this batch-to-batch and line-to-line variability. That's what we're talking about here. And scale-up means we need to create a lot of cells for these models and also for regenerative therapies. So we really need excellent quality control methods for preclinical disease modeling, drug testing, and regenerative therapy in patients. So of course, there's many ways we can check for quality and function of these stem cells. I'm going to cover some of these real quickly. We have flow cytometry. It's great. It's single cell. People look at morphology, so they'll stain their cells and look for sarcomere length and other things that indicate mature phenotype. Gene expression is a favorite method of quality control. You can look at many genes we know are expressed in mature cardiomyocytes. Functional measurements include electrophysiology. Are the cells beating as they should? Are they contracting as they should? The mechanical properties. And the metabolomics, which is closely related to what I'll talk about today. We know the metabolism changes as cells differentiate. So these are great, but they are destructive or invasive. They're low throughput. Some of these electrophysiology measurements are done on single cells, and they're very labor intensive. So that's kind of annoying, right, for lab work. You know, if you're working on a cell culture flask, you have these low throughput methods, you can deal with it. But then as you start to scale up, this becomes a bottleneck, and it becomes a big problem, especially if you want to get to production level systems or even bioreactor level systems, which we would need for regenerative therapy and for really effective disease modeling. So what we're thinking about in my lab is whether we can use autofluorescence to address these problems of quality control in the scale up of stem cell manufacturing. So if we can use autofluorescence, that means we're using a label-free technique. It's non-invasive. We don't have to stain the cells or manipulate them in any way. It allows for real-time monitoring. We can watch them over time. We don't really have to prepare the sample because we're looking at molecules that are already present in the cells. And the cells can remain alive. We don't have to destroy the cells to assess them. And what people in biomanufacturing really enjoy is this closed loop assessment. So we can actually look at the cells within the bioreactor without perturbing them at all, and that maintains quality for use in patients and in disease modeling. So Alina actually talked about this yesterday, so this was handy because I had not talked about stem cells before. She's definitely done lots of work in this area, and she showed the same graphic. So we know that stem cells change their metabolism as they move into a differentiated cell, so a fully functional cell. They move from glycolysis to more oxidative metabolism. Similarly, if we take a fibroblast and we reprogram it, it goes from more oxidative metabolism to glycolysis. So this gives us a clue that perhaps metabolism is one method that we could use to monitor quality as a stem cell differentiates into a functional cell. So what we're asking today is whether autofluorescence can provide this label-free prediction of stem cell differentiation efficiency, which is the specific metric needed for those biomanufacturing facilities. So of course, Alina also covered this very well yesterday. There's been many autofluorescence studies of stem cells as they differentiate. So this is her work, which she covered well yesterday. We know that there's a metabolic shift as hPSCs differentiate into fibroblasts and keratinocyte progenitors. Other studies have used fluorescence lifetime imaging of NADH to monitor stem cells as they differentiate. Also mesenchymal stem cells. This is work from Irene Georgia-Cootie's group. And we also know that you can use autofluorescence and fluorescence lifetime imaging of NADH to distinguish stem cells from feeder cells. So there's been a lot of work out there to really show that NADH lifetimes are sensitive to this process of stem cells differentiating into functional cells. And what we're asking today is very similar, but different in one way, which is that we're wondering if our autofluorescence measurements early in the differentiation process can predict later efficiency. So we're going to test at day one how the metabolism looks in those cells. Are those cells going to efficiently differentiate into cardiomyocytes or not based on endpoint measurements on day 12. And this is what is specifically needed for predictive batch-to-batch and line-to-line variability in biomanufacturing of cardiomyocytes. So this was also covered very well on the first day. Angelica and Marina and Alina and everybody else covered this well, but I'm covering it again. So we know that NADH is involved in numerous metabolic reactions. And what I'm showing here is in the mitochondria and in the cytosol. And the lit up thing here is NADH. So we know that NADH donates an electron to enzymatic reactions to drive these metabolic reactions. We also have FAD and FMN. So Angelica covered FMN very well yesterday. This is mostly occurring in the mitochondria. So what we're looking at first is the optical redox ratio, which is just a nice measurement of the oxidation reduction state of the cell. So we're taking this autofluorescent electron donor, NADH, and we're taking the ratio to the autofluorescent electron acceptor, FAD. So this is really telling us about oxidation reduction reactions in the cell. And many previous studies have shown that this is a great way to test cell health, cell metabolism, malignancy, stem cell differentiation, many things that are coupled to metabolism. So it's a very useful measurement. This was also covered very well yesterday, even by Wolfgang. So we all know that NADH and FAD have distinct states, and their lifetimes are distinct depending on whether they're in a free or a protein-bound state. So here we have free NADH. It self-quenches. It has a short lifetime. But when it binds to a protein, it donates an electron to an enzymatic reaction. It extends, and it has a longer lifetime. So we're looking at two component fits here for both NADH and FAD to account for the free and the protein-bound conformations of these molecules. And of course, many people have been looking at this in many different contexts. It's an incredibly useful measurement for many biological samples. So in our system, we're using 2-photon excitation, and we're doing time-correlated single-photon counting, as Wolfgang explained on the first day. So what we have is our data here in blue, our impulse response function in green. And so for each of these pixels, we have this decay curve, and we're collecting at two wavelengths, one for NADH, one for FAD. And then we integrate this decay curve to get our redox ratio, which is just an intensity measurement. So that's just the number of photons. And as I mentioned before, we have a two-component decay that we're fitting. And then I'm also going to report the mean fluorescence lifetime, which is the weighted average of the short and long lifetime components. So here we're really interested in single cell metabolism because we're trying to predict how those cells will differentiate or not. So it's important for us to then segment single cells. And I think there was a tutorial yesterday on cell profiler. So we're working with the great people at the Broad Institute who have developed cell profiler. And we have our own pipeline here that does the single cell segmentation. So we're segmenting specifically the cytoplasm. And then after we have these segmentation masks for each cell, we have all of our autofluorescent fit variables for NADH and FAD, these summary variables, the mean lifetime, and then the intensities that we use to calculate the redox ratio. So here, this is our study design. What we have is already established in previous studies for stem cell differentiation. So we have on day 0, this Wnt activator that we give, and we actually image right after the Wnt activator has been given. Then we perform optical metabolic imaging, which I forgot to tell you. We just use that to explain all the variables we're taking, which is NADH lifetimes, FAD lifetimes, and then the redox ratio. So that's what that encompasses. We image on day 1. And then there's also another Wnt inhibitor that's given on day 3. We image then as well. And then we image on day 5. And then the endpoint measurement is this cTnT labeling, which occurs on day 12, and positive labeling indicates that those cells have successfully differentiated into cardiomyocytes. So we varied the differentiation efficiency by varying the concentration of this Wnt activator at the beginning of the protocol and by plating density, which has previously been shown to vary differentiation efficiency. We also looked at several different cell lines to see if our measurements were consistent across many different types of stem cells. Okay, so here's the data from day 0 and day 1. So shown here is the mean NADH lifetime. And what we found immediately is that with this Wnt activator, there's a huge change in the metabolism of the cells. And so we expect there would be a big change in the mean NADH lifetime. And what we see is that there's a difference between the low efficiency condition and the high efficiency condition. There's a greater change between day 0 and day 1 in the high efficiency condition. And this is actually true for the FAD lifetime as well, although the changes go in the opposite direction. That always makes sense to me because this is an electron acceptor and this is an electron donor. But similarly, the trend is the same. The more high efficiency condition has a greater change in the FAD mean lifetime. And similarly, for the optical redox ratio, there's greater change with high differentiation conditions, efficiency conditions compared to low differentiation efficiency conditions. So this made us think maybe day 1 was a good time point to predict later differentiation efficiency. I also wanted to show all the data. So I told you we took many variables. So that's what's shown here in this heat map. This is just a z-score heat map, which I've defined here. And I kind of highlighted these really high differentiation efficiency conditions here. The next highest is 38% here. And if we just ask, an algorithm to cluster this heat map, we see that the heat map is clustered by high differentiation conditions and low differentiation conditions. And this is based on all the variables that we take in this study, again, on day 1. So another way to look at this data is to actually do a multidimensional data reduction. So this is called a U-map. So we have all the 13 variables that I showed in that heat map, and it's a multidimensional space, but we want to visualize it on a graph. So the U-map just takes the distance between cells in multidimensional space and projects it onto 2D space, so then we can observe clustering. And the U-map in particular preserves that clustering in the 2-dimensional space. That's why we chose this data reduction technique. So what you have here is the high differentiation conditions. And what we see immediately is that the differences between day 0 over here in black and day 1 in blue are very different. In fact, it looks like day 1 cells are different from cells across the rest of the time course in these high differentiation efficiency conditions. However, that's not the case in the low differentiation efficiency conditions. So in this multidimensional space, they all kind of cluster together, which is one sign that perhaps these cells are not undergoing efficient differentiation. So the ultimate goal was to build a predictive model. So that's what we've done here. We took all the data, all those 13 autofluorescence variables from the lifetimes of NADH and FAD and the redox ratio on day 1, and then we took the ground truth to be the cTnT labeling on day 12. And so here, this is just a binary classifier. So we're just asking, is this a low differentiation efficiency condition? So less than 50% of the cells differentiate, or high differentiation efficiency condition where greater than 50% of the cells differentiate. So it's a binary classifier. And what we see here, there we go. If we use all our variables, we achieve a really high sensitivity specificity. So the AUC is one summary variable, 0.98, that's very close to 1, so that's great. If we just use the NADH lifetime variables, we achieve very high accuracy as well. So this tells me we can use NADH lifetime imaging on day 1 to predict differentiation efficiency on day 12. And I should mention, of course, we use separate testing and training sets. So the cells are separated. We trained on 20,000 cells. So yeah, that's a lot. And we tested on 5,000. And that's where this AUC came from. So I told you only half the story so far, really. So we have differentiation occurring really up to day 10, 12. And that's when spontaneous beating begins. So what you're seeing here is a cardiomyocyte spheroid that spontaneously beats. But what we really need, if we're going to use this for regenerative therapy and disease modeling, is mature cardiomyocytes. And actually, even on day 100, they're not fully functionally mature, but they're definitely more mature than they are on day 10. So the next question we asked was whether we could measure metabolic changes through this maturation process. So we know throughout maturation, the metabolism changes from newborn. So a fetal cardiomyocyte will have a high level of glycolysis, whereas neonatal, it goes way down to about 5%, and we're seeing a lot more fatty acid beta oxidation, which then becomes the dominant form of metabolism by day 21. And I first wanted to prove to you that we actually achieved mature cardiomyocytes. So here is some morphological quantification of alpha-actinin on day 20 and day 150. And we know that these stains should be very regular for a mature cardiomyocyte, and that's what we see. And what we're measuring here is the staining basically on day 20 and day 150 of the surface area, the perimeter, the long to short axis, so that's the structural changes you see here, and the sarcomere length, which is incredibly important metric of how mature these cardiomyocytes are. So if we, I should mention, my scientist Tong Cheng is incredible in the lab, and he was able to pull off this really long-term study across multiple different conditions. It's incredibly impressive. So he was imaging the same cells over this long 90-day time course. So what he has plotted here is the mean NADH lifetime on day 30, 40, 60, 70, and 90. And you can see just looking at this, the cells start to look different. But then if you plot it, you see this really nice continual increase in the mean NADH lifetime over this maturation process. And these differences are significant compared to day 30. So similarly, the redox ratio decreases over this time course, especially between day 30 and the rest of the days. So this is indicative of, a switch away from glycolysis, which is what we expect in those cells as they mature. So overall, that was all new stuff. I love showing you guys new stuff. We found that label-free technologies are needed to evaluate the quality of human pluripotent stem cell-derived cardiomyocytes during biomanufacturing. So our goal today was really to use optical metabolic imaging as a predictive tool to determine what will happen later in these bioreactors. And what we found is that NADH lifetimes on day 1 can predict differentiation efficiency on day 12. So that's a really useful way to avoid wasted time and money in biomanufacturing for batches that are not efficient and batches that will not turn out to be good after several days. We also learned that NADH and FAD fluorescence can monitor metabolic changes with maturation, and we're still analyzing this data, but it's very encouraging. And so we're hopeful that we can use autofluorescence imaging to streamline cell manufacturing from human pluripotent stem cells. So today I talked only about cardiomyocytes, and actually the protocol is different for many different types of stem cells, but it's still a very exciting area. So of course I should mention, I did none of this. This is Tong Cheng right here. He is the stem cell expert. I never would be doing stem cells if he weren't in my lab. I hardly even know what a stem cell is. So he has taken this and really innovated the use of autofluorescence imaging for stem cell research. And our collaborator, Sean Palachek, actually developed this protocol to differentiate stem cells into cardiomyocytes. And what kind of advisor would I be if I didn't tell you a little bit about my students and their talks at SPIE Photonics West, which usually is right after AIM, I know. So we got to wait a whole month, but they're all recorded. So hopefully that means you can take the time to look at what they're doing. And as you can see, most of what we're doing in my lab is autofluorescence imaging of immune cells, T cells, and cancer. So hopefully you can make some of that. But for now, I'm happy to take any questions.
Thank you, Melissa. Okay, so we have a question from the chat. Do you see any use for such tools in flow cytometry?
Yes, it's so funny you asked that because we're in our, well, that's one of the talks actually at SPIE. We're building a flow cytometer for T cells, and that's for T cell manufacturing, which is a CAR T cell therapy thing. And what we're finding is that we can also measure fluorescence lifetimes very quickly and even fit decays quickly in a flow geometry, which is pretty great because those cells are flowing really quickly. But I'm finding that this is what people really want because actually they want to be able to sort cells. So they want to know based on the lifetime, can we get pools of different metabolic cells and then analyze them more deeply, because we're finding that that NADH lifetimes are separating cells that aren't separated clearly with other methods. And we're really curious about what differs between these pools of cells. So I'm super excited about flow cytometry, yes.
Okay, and then we had another one. Did you compare the different biomarkers with other readouts, metabolomics or staining?
Yes, and I didn't show that. I think I have an extra slide. I could probably show it. But we looked at really simple glucose and lactate assays. And we know from previous studies that the metabolism should change, as cells differentiate. So if there's a low efficiency condition and a high efficiency condition, we should see changes. So this is just a plate reader measurement of lactate and glucose in the media under high efficiency and low efficiency conditions. And actually, our collaborator, Sean Pelichek, is a metabolomics guy. So he has done a very extensive study of the media conditions. And this is just 2 metabolites, but it confirms what we expect.
Okay, and then one last question. Are there any other biomolecules other than NADPH, FAD that might contribute to autofluorescence? What is the possible effect of oxygen, CO2, etc. concentrations in the culture media on stem cell differentiation and fluctuations in lifetime?
Those are great questions. Yes, lipofuscin is a guy who hangs out in there. Flavins, there's many types of flavins. So there are other fluorophores contributing, and it could be in the FAD channel that we're seeing those. Definitely oxygen, CO2, and media conditions affect stem cells. I've never seen cells that were so finicky. So if there's any tiny little change in the buffer, they die. So it's really important to monitor these metabolic changes, especially if you're trying to scale up.
All right, so I think we're ready. Thank you for a great talk. I think we're ready to move on to the next speaker, and there's a couple more in the chat there.